Dear students, Assalamu alaikum. It's Dr. Sibtan Ahmed and I'm working as assistant professor at University of Agriculture, Faisalabad, Pakistan. The course I have been assigned is MB402 and the title of this course is Basic Microbiology, and this course comprises of 3 credit hours. Out of these 3 credit hours, 2 credit hours belong to theory and 1 credit hour belongs to practical. In this video, I would be discussing with you the contents of this course along with the recommended books. As I already told you that this course comprises of theory and practical. The theoretical portion include introduction to microbiology, and in this topic I will be discussing with you scope, definition, branches and applied areas of microbiology. The next topic is historical introduction, including work of Pasteur, Koch, Lister and recent other developments in the field of microbiology. In the third topic, we would discuss diversity of microbes, differentiation between prokaryotes and eukaryotes, taxonomy of bacteria, structure of eubacteria, and in these different structures include the nucleus, flagella, fimbriae, the bacterial endospores, and cytoplasm. After this, the topic of microscopy would be discussed in detail. In microscopy, we would be discussing light, dark field, fluorescent, polarizing, phase contrast, and electron microscopes, which are different types of microscopes. After this, you would be familiarized with dyes and staining, light and color, absorption spectrometry, general chemistry of dyes, indicators, nature of staining processes, factors influencing staining, and staining of living cells, determination of isoelectric points, and cell structure by staining, along with different types of stains. Later on, we would be discussing brief introduction to structure and propagation of fungi, protozoa, algae, viruses and bacteriophages. And lastly, introduction to biofertilizers and biopesticides and the application of different microbes in food industry and use of these microbes in bioenergy and biofuel sector and in drug industry. Practical portion would start with the topic safety rules and principles while working in microbiological lab. Then different lab equipments would be showcased which are being used in the field of microbiology. The third topic is microscopy. In this topic we would cover principles and types, use of bright field light microscope, also preparation of slides for microbiological examination, simple staining for bacteria and fungi, gram staining, contaminant and decontamination. Then sterilization and disinfection, which includes physical agents and chemical agents. Evaluation of antimicrobial activity, culture media and its types, bacterial or fungal cultivation, bacterial or fungal colonies, and in this topic we would be discussing the types and characteristics of bacterial or fungal colonies, bacterial or fungal morphology, and by morphology we mean that we would discuss shape and arrangement of bacterial or fungal, then micrometry, and lastly bacterial motility demonstration would be accomplished. And recommended books for this course include Collins and Lyon's Microbiological Methods, Medical Microbiology, Brock Biology of Microorganism, Topley and Wilson's Microbiology, Mechanisms of Microbial Disease, and Foundations in Microbiology, and lastly Microbiology, an Introduction. These 7 books have been recommended for this course. Thank you for your patience and listening. Stay home, stay safe, prevent corona. Thank you very much.